And with that, I think we can move to start. So with that, from my end, again, welcome to the first SentroConnect live webinar. It's great to have you all joining here. We'll have a number of presentations, 2 in total, from the Centronic team. And with that, we'll start in a moment. For the time being, I do want to quickly comment on the poll. For that, I will now end the poll to be sure. Good. And you should all see the results already. So as we can see, we do have quite a number of continents present. We have a larger, or the majority from Europe, but also from North America and South America, as well as from Asia. I think that does pretty much fit the registration, Sebastian, from what I can tell. The SentroConnect Live is targeted not only as experienced, but especially at novice industry members for PAT. Therefore, I'm quite happy about the results. So we do have, of course, a number of SMEs, all in PAT pros and chemometrics, but we also do have a representative number of basic experience or not yet PAT practitioners. So especially those who might be interested at learning the basics of PAT here, specifically NIR and the analysis. So with that, I'm quite happy to see that on the result, which fits the target audience as well. And with that now, it's my pleasure to introduce the team. By the way, my name is Hubertus Rehbaum. I will be just moderating today, not presenting, but instead we do have 2 presentations. However, before we jump into the presentation, we want to get to know the team from Centronic, both in Germany and US. And with that, I will give the word to Uwe to start with an introduction and then do a round table.
Okay, thank you, Hubertus. Thank you very much also for organizing and helping us to set the first SentroConnect live up. Great and good experience to work with you, as always. So I'm Uwe, co-founder of Centronic, and since 1993 with Centronic, I came from Technical University in Dresden with an electrical engineering background, and my work life is Centronic since then.
Great. Then we'll go on to Owen.
Hi, I'm Owen Rehrauer. I'm the president of Centronic US Corp. I've been with the company since its inception in 2022, although I've been a global Centronic partner since 2019.
Right. Sebastian?
Hello, everyone. My name is Sebastian Zawinski. I'm with Centronic since 2000, and I'm key account manager at Centronic GmbH in Dresden.
And last but not least, Adam.
Hi, everyone. My name's Adam Risch. I've been with Centronic US as a scientist since last year. So about a year and a half with the company.
Great. So the focus of today will be both Owen and Adam. Today's first webinar provides introduction to PAT and data analytics. So especially from the US team, those 2 will be presenting. Of course, we are looking forward to your questions. So any questions that you have, you can already provide or enter in the Q&A system of the webinar. However, we will only move on to answer the questions after both presentations have been heard, just that we will allow to have the full presentation and then sufficient time for answering all of the open questions. The first presentation will be by Owen to investigate and understand NIR PAT for bin blending. So that is focusing primarily on the technical hardware. Afterwards, Adam will speak about chemometrics with NIR spectra, both providing an introduction. So really this webinar is also intended for novice PAT practitioners and those who want to learn about this. And with that, I think we can start now and move on to Owen. I will, with that, ask Owen to start sharing his screen and look forward to your presentation.
I apologize, Hubertus. We seem to be having some technical difficulties. I'm unable to share my screen.
Yeah, that was me. I should, of course. Now you should be able to do that.
Fantastic. Thank you.
You know, if it's technical, I'm usually at fault.
Shucks. Well, apologies for the technical difficulties here, folks. That having been said, thank you all for joining the inaugural SentroConnect presentation. As Hubertus mentioned, my name is Owen Rehrauer, and today I'll be presenting to you a basic overview of PAT and NIR PAT with a focus on bin blending applications for pharmaceutical analysis. So we're going to start with some real basics. What is PAT? Why people might want to use PAT, particularly in a pharmaceutical application. We'll cover some of the fundamentals of NIR PAT from an instrumentation and application perspective, and then spend some time on solutions and some technical aspects of bin blending for pharmaceutical PAT. What is PAT? I've got the definition that the FDA has put in their PAT guidance up on the slide. I'm not going to read off the slide, but I want to highlight, and I've done this via the bolding in the text here, that this is timely measurements made, for example, during a process that are tied to critical quality attributes that help ensure final product quality. That differentiates PAT from other soft sensor measurements. What's motivating the use of PAT? We've used conventional laboratory analysis for many years successfully, and people may be asking why change for something that's been working well. Well, there's a long time between the actual processing in pharmaceutical manufacturing and laboratory analysis, and that unfortunately prevents the ability of making interventions if a process is undergoing a change from its target performance. The cost of losing a batch as a result of this can be quite high, so there's some business incentives to want to use PAT to avoid this eventuality. Sampling material can also be challenging. If you're taking something like core tablets from a tablet press, that's quite easy. But thief sampling and bin blending, for example, is known to be prone to sampling errors and problems that then create investigations, deviations, and just generally some headaches. So if we can avoid that, there's also some good reasons to do that as well. In the US context, the FDA also will, in some cases, give advanced manufacturing designation to control strategies that include PAT. So there can be some regulatory advantages in terms of speed of filing and approval of a drug via the use of PAT. And last but not least, these measurements can be useful in process understanding and optimization, particularly during product development. So what are the benefits? Well, there are going to be answers to all of those, but there's a few others I want to highlight as well. So yes, of course, PAT can get you real-time process insights. That can be used as a guardrail to help avoid any failures of batches, but it can also be useful for making interventions or getting some understanding of the process itself. When implemented in an inline or online way, there's no sampling required, so we can get away from things like thief sampling. In addition to the benefits and motivations I mentioned before, I do want to highlight that PAT can be useful in implementing a quality by design or QBD approach and can be used to support real-time release testing. QBD is something that I find a lot of novice PAT users have some confusion on. In fact, some people think QBD and PAT are synonymous. So I do want to just cover what QBD is and how PAT interacts with it. When we look at conventional manufacturing, how we've done pharmaceutical manufacturing for quite some time, we have to acknowledge that there's some variability in the inputs to our process, whether that's material variability or environmental variability, etc. Historically, we've tended to use fixed process parameters to run a process, so a constant speed, a constant compression force, etc. And from that process, we'll get a variable output. We test at the end, and we hope that we're within an acceptable range of quality. This, of course, is not ideal because occasionally we will not be within the range we would like for quality, and we have to reject some material from use. QBD manufacturing, in contrast, of course, starts with the variable inputs. However, we use a design space of acceptable ranges of processing parameters. So rather than having a fixed number for speed or compression, we, during development, would qualify an acceptable range of speeds, compression forces, that we can use to get acceptable product quality over the range of variable inputs to our process. And then we adjust our processing parameters within our design space to help ensure consistent product quality. By using PAT, we can make measurements to best understand where in our design space we are and where we might want to set our processing parameters to help ensure that high product quality. It's often said that PAT enables a QBD approach. This is how PAT does this. I just wanted to cover that basic point because I do see a lot of questions on it and hopefully preempt those from the chat. That's an overview of some of the fundamentals of PAT, but let's talk about NIR PAT in particular. Here, I have a cartoon version of an NIR spectrometer and associated probe system. I want to cover just some basics of the instrumentation so people have an understanding of what's going on under the hood inside the PAT boxes that you'll actually see on the floor. In its most basic form, NIR spectroscopy is an absorbance spectroscopy. Oftentimes in the pharmaceutical or food contexts, we'll use what's called diffuse reflectance spectroscopy, where light's shown on a sample, and we look at the light that's back reflected from the sample itself. This is in contrast to transmission spectroscopy, where we'd actually shine light through a sample. That can be challenging with things like powders and tablets, given the thicknesses we typically work with. So light is going to come from some broad light source. Usually it's transmitted through a fiber optic cable into something like a process probe or to a process window. We measure some light, which comes back from that sample, and we transmit it to a spectrometer. Centronic tends to prefer using diode array spectrometers. So here I've shown a cartoon diagram of what that looks like, where light is transmitted to a grating. That grating separates the light based upon its wavelength, and then it's detected on an array. The output of the detected spectra is then transmitted to a computer where it's saved to file. This is how the magic happens inside the box. I do want to cover a little bit about diffuse reflection. As mentioned before, we're looking at light that's back reflected from the sample. We will typically, at the beginning of an experiment or the beginning of a day, measure what's called a 99% standard. Some people may call this a background, to understand how much light can be reflected when almost all, 99% of the light is back reflected. This helps us get an absorbance calculation. When we measure this in powders, though, I want to highlight that the light is going to penetrate somewhat into the sample, getting deeper and deeper in, depending on some of the sample qualities. These can differ based upon whether the sample is more or less dense, or its particle size distribution has changed. This affects the functional path length, how deep the light goes in and comes back. And as a result, it's often said that diffuse reflected spectrometry includes information about chemical as well as physical sample information. I believe Adam will talk about this later because for some analyses, we want to avoid information about the physical attributes of our sample. In others, we may care about that quite a bit and use different analysis methods to highlight that information. So what are we looking at when we look at an NIR spectra? We are looking at harmonics and overtones of the mid-IR spectra. Here I've shown the near-infrared spectral range and the chemical moieties where these different functional groups will have absorbances. Unfortunately, because we're looking at harmonics and overtones, it can be a little bit difficult to do something like structural elucidation from the NIR spectra of a pure sample alone. We instead get something that can be thought of more like a chemical fingerprint. Here, I've shown the NIR spectra over a range of water. I do want to highlight for powders that there's a wavelength-dependent depth of penetration. How far light goes into a sample can depend on wavelength. And I also want to highlight in general, as we move from 750 to 2500, the intensity of absorbance bands tends to increase for some basic physical reasons. Here, I will also highlight for the system that we're going to talk mostly about today, the spectral range where we're looking. Our systems tend to cover the majority of this range. We don't tend to cover all of it as a Fourier transform system might. This is a trade-off we make to ensure that we have the most robustness possible for a process area, and so we're robust to things like vibration and temperature changes. I also want to highlight that our systems cover the range of 1700 to 1900, which can be very important in a lot of pharmaceutical applications, as this is where many important peaks from things like lubricants can occur. What might someone use NIR-PAT in a pharmaceutical application for? Well, common applications include monitoring blending uniformity during mixing, whether that's bin blending, as we'll discuss more today, or for continuous blending and things like continuous manufacturing applications. NIR-PAT is also routinely used for content uniformity. This can take the form of, for example, transmission NIR measurements of tablets. NIR is commonly used for material ID, so looking at raw materials before they're placed in the process area to confirm that when we think we have microcrystalline cellulose, we really do. And of course, NIR is commonly used for drying applications. In pharmaceutical applications, this could be something like monitoring fluid bed drying. Sentronic provides a number of solutions for these different applications. Here I've highlighted different common unit operations in pharmaceutical manufacturing, and just wanted to quickly mention that we have solutions for a variety of these. The orange checkmark, which you'll see at the bottom right-hand side, is our bin blending application solutions. We're going to be talking about the system on the left, the smaller SentroPat DA, although I will note that since 2005, we have been providing solutions for bin blending. The larger SentroPat BU2 is a venerable system that's been used for quite some time, whether it's in development or in commercial applications. Last but not least, we do have a newcomer to our offerings, the SentroPat ID, which can be shown here, which can be used for identification of raw materials. You should be hearing more about this product from us shortly. So you might be asking, why would someone want to use NIR-PAT for bin blending? Well, we can avoid thief sampling, which tends to be a very desirable thing for a lot of people that do bin blending in pharmaceutical applications. We can also provide some information about the potency of the final blend, which can be useful for understanding the success or failure of following tableting. It supports some data process optimization. So if we are interested in optimizing how many revolutions we execute, we might take a guess that we need to do 300 or 400 just a priori to make sure we really get to blending, and then find out via NIR measurement that actually after 150 revolutions, we're actually blended. If we're making multiple batches per day, shaving off 80% of your total blend time can be a big win from a product efficiency standpoint. Last but not least, the NIR-PAT can be useful in troubleshooting root causes of unusual product behavior. We can be sensitive to things like segregation and agglomeration in ways that may even be difficult to find from thief sampling. As a result, there's a lot of measurement benefits to using NIR-PAT, and we're seeing more and more pharmaceutical companies get interested in using this for bin blending. So how does this all work? You're going to have to I apologize for my poor artistic skills, but this is my graphical representation of the bin blending process. Here in the middle of the circle with arrows around it, you can see my representation of a bin with one of our SentroPat DA systems stuck to it. We'll put the system onto the bin, and then we'll start rotating the bin around on its rotating. As we move around, we will come to a predefined angle, at which point the NIR-PAT system will make a measurement. We use an internal 3-axis accelerometer to detect the angle of the system and trigger a measurement at a fixed angle. We do this because gravity will force powder to a process window we've installed on our bin that connects to the NIR-PAT system. And when powder's in front of the window, that's an ideal time to make a measurement. Suffice to say, if there's no sample in front of the window, we're not going to get a lot of process information about this. We have found that trying to do this via timed measurements as opposed to actually using the angle itself can be tricky for synchronization, particularly for bin blenders that actually will change the direction of rotation. Timing can be quite difficult there. By using the angle, we seem to get the most robust sampling of our material. So we're going to make a measurement as we're going around, and then the bin blender is going to continue to rotate around again and again and again, collecting one NIR-PAT measurement per rotation. When we're done, we'll stop our bin blender, we'll stop our PAT measurement, we'll take our system off, and then we'll go to analyze the data. How do we do that? Well, Adam will cover that in just a few minutes. This is all well and good, but let's actually take a look at this on a system itself. So here I have one of our systems installed in our unit operation simulator, the Centro Blender. We have a glass vial here so we can see quite easily when measurements are made during the rotation process. So we're going to start rotating the Centro Blender, and you will see when powder is in front of the window, we turn the light on, which is indicative of a measurement, and we turn it off when powder is not there. So on, off, on, off, and again and again and again. So what does that process window look like? Here is a picture of a modified bin lid, which tends to be the most common way NIR PAT is connected to a bin for blending. We have, of course, also modified the bins themselves, but by modifying the lid, we can use that what was commonly called a PAT lid with multiple blenders. Multiple bins, which can be quite useful if we're comparing or trying to scale up different bin sizes. On the other side of this will be a flange, and that flange can be used to, using a C-clamp, attach the SentroPat DA. So speaking of the SentroPat DA, this is our latest and greatest solution for bin blending. It covers a wide range of the NIR spectrum, allowing us to be sensitive to most APIs, excipients, and lubricants. It has internal intensity and wavelength standards, which can be quite handy because you can use these for system suitability testing, making sure the system's in good health, without having to have operators bring the standards onto the process floor. For bin blending applications, we want to, if we possible, be completely wireless. It would be a bit challenging to connect wires to a moving bin, so this system is fully battery powered with a long battery lifetime, as well as fully Wi-Fi enabled, so we can communicate with a laptop or local server without the need to connect communication cables to the system. It's also quite lightweight, weighs about 5 pounds, and it's small enough to fit on development blenders like V blenders, but also still is quite useful on commercial scale blenders as well. In terms of control of the system, we'll touch on this very, very briefly, but we have an internal set of software called SentroSuite GMP, which can be used for control and a collection of measurements on all our systems, including the SentroPat DA. This software can also be used to execute, common validation tests. These tests include tests tied to the USP 1856 as well as EP 2.2.40 test requirements, as well as some vendor specifications as well. And the software is, of course, compliant with the relevant versions of 21 CFR Part 11, which is quite important for our pharmaceutical customers. In addition to this software, we also do have the ability to control our system using some common control softwares. Things like Siemens SciPat, SciY, SyntaQ, etc. If you have questions about this, feel free to reach out and we can talk about what we're compatible with. So what are some common integration questions that we get? We get a question a lot, can the system be controlled via OPC communication for integration with blender control software? The reason for this question is commonly because people would like to use an endpoint detection determination made via the PAT data. Basically, once we get to a certain point, can we have the NIR tell the blender to stop blending? The short answer is yes, we can. If you have additional questions, feel free to reach out. There's a question often around what flange sizes that our systems are compatible with. Commonly, it's 2-inch, 4-inch, and 6-inch diameter flanges. These allow us to be compatible with the Centronic historic PAT solutions for bin blending, the SentroPat BU2, as well as several other vendor solutions. Again, if you have questions, feel free to reach out. And we, of course, always get questions. It's a real common PAT one. If we take laboratory analytical systems, bring them to the process floor, are they going to be rugged enough? We have spent a lot of time ensuring our systems are rugged enough. However, I would recommend avoid doing something like this. These are tough, but try not to hit them with a forklift if you can. So let's put it all together. We've mentioned this is complicated, all the different steps involved, but I think you'll see in the attached video where I believe one of the members from the Centronic team here is going to be actually showing you how easy it is to collect NIR PAT data. Oh, here we are. Sorry about that. So you can see here, Sebastian is attaching the system to its PAT lid. We're going to then connect the bin to our blending arm. Slowly move up. We're rotating around, collecting our blender revolutions. You can see our system there. And from that, we're going to get spectra in SentroSuite. That's as easy as it can be. With that, I will say thank you for everyone's time, but more importantly, I want to pass it over to Adam for his portion of the presentation. Thank you for attending, and I look forward to your questions shortly.
Perfect. Thank you, Owen, for the presentation. A brief reminder to everyone, of course, you can already put questions now. Note that the questions, you put them down in the Q&A system, will not be visible to everyone, and I would like to encourage you The webinar is meant to answer any questions you have. There are no basic questions not allowed, so feel free to share them. And you can also ask anonymously if you prefer this.